are both postdoc at the Epilepsy and Brain Network Lab at the HUG under the supervision of Serge Vouillemont. So just a quick outline before we start the tutorial. The tutorial will be split in 4 steps, where we will cover the main steps to do electrical source imaging. The first part I will introduce the method, and then Nicolas will show you the practical scripts that we implemented on Matlab. So the first part will focus on the pre-processing of the functional data, so on the pre-processing of the EEG. So we will have a look at how to import the data, filter them, identify the bad channels, remove the artifact, and re-reference to the average. Then on a second step, we'll have a look at the anatomical data. So we'll go through MRI pre-processing to compute the head model and source model that are necessary to then estimate the forward model, or the so-called lead field matrix. This will be seen in a third step, where we will propose the solution of the inverse problem. So we will go through the lead field matrix extraction and the spatial filter estimation. And then in another step, in the last step, we will basically go from the time course at the source level to the time course at the brain region level. Before going through the EEG preprocessing pipeline, just a few words on the stimulus that we are working on. The functional data are visual evoked potentials. So basically the participants are presented images of faces and scrambled faces that are basically obtained by just phase spectral randomization of the original face images. Each participant is presented with 4 blocks of 150 trials each, where 300 trials are faces and 300 trials are scrambled. So regarding the EEG, the first step of our pipeline is again about our functional data, so how to clean the functional data. So the data are recorded with a 128 channel biosemi active system. So the data are actually expressed as voltages between each electrode and another electrode that is called the CMS, or the common mode sensor, which contain the 50 Hertz noise. So in order to get rid of this contribution, we need to re-reference the data to one channel. And in this case, we've selected a central channel A1 that corresponds to the CZ. And this can be done simply with the function proposed by Filter that is called FT preprocessing. Later on, to reduce the computational load, because of course we are working on continuous data, we downsample the data to 250 Hertz with the function FT resample data. And then we again use this really useful function FT preprocessing to high pass filter above 1 Hertz. At this point, we do not really mind about eye blinks or other artifacts that will be later on identified by independent component analysis. Since we're working on continuous data, and of course we want to focus on evoked potential, we need to segment the data. And in order to do this, we go and read the file that is called events.tsv file that you find in the dataset, where we have the onset of the trial plus the value. The value is simply a flag of 1 corresponding to faces and 0 corresponding to scrambles that identify what were the trial that the participants were proposed. We then define and this can be done with the function ft read event, where file name is simply the name of the event file. Then we define the epoch of interest. So we define how much of pre-stimulus we want and how much of post-stimulus we want. And we simply segment with ft trial fungen, which is a low-level function of field trip. And we actually then segment the continuous data into epochs with ft redefined trial. Once the continuous recording is segmented, we need to identify the bad channels. And by bad channels, we mean all those channels that have a low signal to noise ratio. That can be due, for example, by high impedance because of a bad contact. They can be identified in different ways. In general, a channel that, for example, has a low correlation with the channel next to it can be a channel that could be labeled as a noisy one, or a channel with a standard deviation that is very different, that exceed the standard deviation of the other channels, could be as well defined as a noisy channel. In our case, the bad channels were already identified by visual inspections of the epoch, and are stored in the file that you find in the dataset that is called channels.tsv. So what we do, we simply read the file from the dataset, and then we keep the good channels with FT select data. Once the bad channels are removed, we run the independent component analysis to remove the artifacts. So the independent component analysis is a blind source separation method that basically allows us to separate signals that are actually originating within the brain from signals that might be originated from other sources, for example, like eye blinks or muscle artifacts or external noise. Different algorithms exist. Here we calling FT component analysis, which is the function through which we run the ICA in field trip. We use the method Tunica, which corresponds to the Infomax used in Eeg Lab, if I'm not wrong. This step needs, of course, to be done after bad channels removal, because we want the results to be, the signal to be least corrupt as possible in order to have results that are easy to interpret. The results of ICA yields components that are independent from each other and are non-Gaussian. We can inspect these components with a really useful function that's called FT data browser that allow us to scroll through the components and the epochs. Here, for example, we see the first component is quite clearly different from all the others. We can clearly see that this is not a genuine EEG signal, but more likely to be eye blink, because of course, the very low frequency content of the signal, because of the topography of the signal, and because of the amplitude that, of course, is bigger than that of the other channels. So once we have selected the components that we believe not to be of genuine EEG signal, we reject this component with FT reject component, and this basically back process the bad components to the sensor space and then subtracts these bad reconstructed channels from the original data. The last step is then, of course, to replace those channels that we defined as bad channels and that we removed from the dataset. So first of all, we need to define what is the net of neighboring electrodes. And in order to do this, we can use FT prepare neighbors. It's simply defined based on the layout of our electrodes, which electrodes are relatively close to each other, depending on either a triangulation method or a distance method, for example. And then we simply use FT channel repair to interpolate the bad channels with the signal of the surrounding electrodes. And this can be done by for example, by just taking the average of the surrounding electrodes or the surrounding signals, or by means of spherical interpolation of the neighbors. Here, for example, we have the first 11 channels. Of course, the first one is flat because we have re-referenced our data to this channel. And then we have the A1, A9, A10, A11, which are clearly noisy. After the interpolation, that's the results. The last step then is to re-reference all our channel to the average. So basically, the average of all the channels is calculated and then is subtracted by from all the different electrodes. And this is needed for source localization, so that the error present in the forward intrinsic to the forward model can be averaged across all the electrodes. And this again can be done with FT preprocessing. Now I'll pass the stage to Nico, who will show you how to do this directly in Matlab.
Right. So here I'm presenting a Matlab live script. And I don't expect you to follow and do it on the virtual machine with us, because most of them are taking some time. So basically just follow along, and afterwards you can play with modify the parameters and so on to see how, for instance, moving a given ICA impacts the data, or changing some more model for the forward model impacts also the data. And as Seb said, we had to do a modification, so please do a git pull before launching everything. And again, if you want to relaunch everything, you should better use the script and not the live script, because this is more for the presentation. Okay, so the first thing is to basically set the path in Matlab and set the filter path. Also, you should never put all the folders of the field trip toolbox inside the path, but just put the main folder and then do FT defaults, because otherwise it will fail. All right, so yeah, basically here we do some initialization. Again, the downsampling, as basically as Isotta already explained. With downsampling, we read the data, we redo the preprocessing, so the re-referencing to the A1. This is done just because it's an active recording system. So most of the AEG recording are not active, so you probably don't need to do this step. So this is specific to this data set and to the biosemi recording system. And okay, blah, blah, we do the events. We remove the bad channels. So again, as she said, we read the bad channels from the channel TSV file. And here, if we plot the bad channels, you see, here I put a bit more, and you see that, for instance, like they have a lot more high frequency content than all the others, especially, do you see the, yes, you see it. This one is quite different from its neighbors. So this is clearly a bad channel. Okay. So we remove them from the EEG coding. Then we did the ICA using this, but here again, it takes some time. So we just load, preload what we already computed. And now we can look at the traces. So again, here you see something with very high amplitude and very low frequency. Clearly, the map shows that the dipole is facing, it's going like that. So it's a night blink. And basically, this is what we will remove from the data. Okay, so if we do that, and then we compare what we have removed. So if I do this like that, and this like that, so you see here, this is the raw data, and here is after the eye movement, so the ICA removal. If you use this topo plot from field trip, you can see that it's the same topography as the ICA. And indeed, if we remove the component 1, which is related to the eye blink, you don't see those anymore. And the topography is obviously also different. So really, it's a nice thing to just look at the traces to verify that everything went smoothly. Here, yes, we will also, like, as Isotta said, interpolate the bad channels, but first we need to define a layout and neighboring channels. So here, basically, you see how the different electrodes are connected to each other spatially. Then we use that to interpolate the channels. And when we visualize that, you see that, so again, here and here. Okay, you see here again, for instance, here, this one, we have very high amplitude, high frequency content, but it's not there anymore here. So this worked. So now we have basically clean data. And final step then is to do the referencing here as written, and we can go on with the next step. Yeah.
So once we are sure that our data, our functional data are as clean as possible, so as free as possible from artifacts, we can focus on the anatomical data, so on the structure. First of all, we need, for the solution of the forward model, so the model that describes how the signals originating at the source level is seen at each electrode on the scalp. We need, of course, a description of the different layers of the different conductivity in the head. So for this, we need the head model. So the head model simply is exactly this, a model that describes the electrical properties of the head and is necessary to solve the forward model. In field trip, we can estimate it starting, of course, from the structural MRI. So first of all, we need, of course, to import the T1 with FT read MRI. Then we segment the MRI to extract the surfaces of the skin, the skull, and the brain. More tissues are available in field trip, but for us, these three were sufficient. And this can be done with FT volume segment. From these surfaces, we create 3 meshes using FT prepare mesh. And then we use a head model made of 3 concentric spheres. So basically, one single sphere is fitted on these 3 layers to define the common centers. And then starting from these centers, 3 spheres, concentric, are fitted on the 3 different tissues. So on the brain, on the skull, and on the scalp. And this can be done with FT prepare head model. Here in the corner, you see a plot of the 3 spheres, concentric, with the 3 different values of conductivity for each layer. Once the head model is defined, we can work on the source model. The source model simply is a model that describes the position of the sources in the brain. Of course, the sources are placed directly in the gray matter. And to do this, of course, we need a description of where the gray matter is with respect to our T1. In our case, we place dipoles, current dipoles in the gray matter after the removal of the basal ganglia. And as Peter said, dipoles can have a fixed orientation or an unfixed one. Usually the fixed orientation is defined normal to the surface of the cortex. In our case, we opted for an unfixed orientation. So for each point source, so for each of these points that you see in the grid, we have actually 3 dipoles, 1 for each axis. And this is as well necessary to solve the forward model together with the head model. In order to estimate this, we need, again, to import the gray matter mask and the atlas. These are output of the connector mapper. And this can be done, again, with FT read MRI. We then need to use the information, so the labeling from the atlas, to remove those voxels that correspond to the basal ganglia from the gray matter mapper. And then we simply co-register the gray matter mask and the grid imposed within the mask with the template to have a correct grid orientation, so that we have a symmetrical distribution of the source point between the left and the right hemisphere. And then with FT prepare source model, we impose that a 6 millimeter resolution grid is defined within the gray matter map. Yes, okay, so this is the end of the anatomical data processing, and now Nicolas will show you how to do it practically.
Okay, so yeah, so basically we have to load all the MR images we need, the classical MRI, so anatomical MRI, gray matter, and the atlas. So to do so, just set the path, you ask field trip to load everything. Here, yeah, there is a little thing, is that for some reason, I don't know why, but field trip does not manage to read directly the compressed images, so the .ne.jz. So we have to decompress it manually, but otherwise you can just open the Nifty file with Filtrip and display those. So Filtrip also can display some, the anatomical images. And basically here you see different slices of the MRI, the gray matter mask, and finally the atlas. And the atlas that you are using is the output of the connector mapper 2018. Wait, I cannot reach the. Can't remove that thing.
Just like from here.
Yeah.
Yeah.
Okay. So yeah, as you see, you have the different paths labeled and so on. And obviously the basal ganglia are removed because we don't really want to have sources there, because we know that the stimulus are not supposed to have any response there. Okay, so that's what I did. Then here, as Isotta said, we had to segment the MRI. The thing here is that because it's an open data set, basically the data were defaced. And that means that you have holes. So we had to do some trickery to make meshes complete. And that's actually a very important point, because if the mesh are have holes, any model won't work, and especially the boundary element model won't work. So basically what we did here is that we dilated the skull so that it will replace the part of the scalp that was missing. Also, unfortunately, it didn't work. So we'll see afterwards, but it's a good way to see that actually the BEM model are a bit tricky, because sometimes they simply don't work because they are very sensitive to the MRI, and sometimes, the classical BEM-CP method of Filtrip don't work, and you will see. And that's why we use the concentric spheres. And as Isotta said, basically, you just have a sphere and then you have different layers and so on. Okay. And yes, here you have the mesh. So as I said, that's annoying that I have the thing in the middle. You see that now it's not open anymore, so the scalp is closed, thanks to our little trick. And you must be sure that everything is closed, otherwise you will have kind of spikes, or in Filtrip it's called star-shaped mesh, and you will have some issues. Also, you must be sure that the different layers are not intersecting. So for the spherical model, it's fine, but for the BEM and the FEM, finite element model, you will have issues. And you must be sure that they do not intersect. And if it's the case, you will need to either smooth the segmentation or play around with like im erode or no im close, so you close the image so that there is no hole anymore. And also make sure that there is a good amount of distance between the different layers, otherwise they could fail. Right, and the source model, as Isotta said, basically you load the gray matter map, you remove the basal ganglia. What we do on top of that is we realign to a template. And this is also linked to the paper that Peter this morning talked about, the one from Christophe Michel and Denis Brunet, where they said that you need to realign to a template. Otherwise, you could have a grid that is not aligned to your head, and then have basically a part of the brain that have less sources, and then you will have weird representation. Okay, so if I plot that, and I open it. Again, here you see that it's, basically you see that you have a line, so it's basically symmetrical. And if we didn't do this step, so it happens especially when you have clinical data, if you have a kid or whatever, and the head is not really centered, you could have like a grid that is weirdly shaped if you don't do this step. Here you can also verify, I don't know if it's very visible, but here you have holes, because we don't put sources in the ventricles and also in the brain, in the basal ganglia, and the cerebellum as well. Yeah, basically the wave pattern, indeed. And final step is to verify that the Eeg electrodes are aligned with the scalp. And here we don't show how to do it manually, but there are lots of tutorials to do that. But we basically read that directly from the beads data set. So it's the electrode TSV file, which is already co-registered with the scalp. And I think that's it for me right now.
Yes. Okay. So once we have computed our head model, source model, and we're sure that the location of the sensors is co-registered with the anatomical data, we can proceed with the extraction of the lead field matrix. As we said before, let's just have a look at this equation. In this equation, A is the lead field matrix, S are the signals at the level of the sources, and X are the signals at the level of the scalp. And basically the lead field, and N of course is the noise. A basically is the model, is the matrix that describes how we go from sources to scalp. So how does a signal in a specific source point, how is it read, how is it projected at the level of the scalp for each scalp electrode? Of course, in our case, for each source, so for each point in this grid, we have 3 currents dipole. We have one in X, Y, and Z. So, of course, the lead field matrix will be an X by 3 matrix that represents the contribution of each of these 3 current dipoles of each of these sources to each of the electrodes. And of course, in order to understand how the signal, how the current travels from the sources to the scalp, we need to, first of all, know where the sources are. And this is given us by the source model. Then we need to know how the signal travels through the head. And this is given us by the different conductivity layers, the different conductivity values in the head model. And of course, we need to know what is the position of the sensors respectively to each other and to the source itself. And this is given us by the actual location of the sensors co-registered with the MRI. Once we have all this information, we can estimate the lead field with FT prepare lead field, a function in field trip. So once the forward model is solved, so once we know how to go from sources to channels, let's say, we have to solve the inverse model. So we have to work out how to estimate W. In this equation, again, X are the signals at the scalp level, S are the signals at the source level, and W is our inverse operator or the spatial filters. Basically, W is a set of spatial maps that tell us how to go from the channel level to the source level. And of course, we have one not only per source position, but one also per source dipole component. W can be estimated with imaging approach, so by the inversion of the distributed source model, the equation that we saw in the slide before, like for example, the minimum norm estimate method, or with scanning approach like beamformers. It's important to say that the solution is ill-posed, which means that the sources that we're trying to estimate, the number of sources that we're trying to estimate is much, much bigger than the number of variables that we have. How do we estimate this? We use Iloreta, which is an M&E-based inverse solution that adopts an optimal weighting method for deep sources. to obtain, as Peter said, spatial smoothness. And this can be done directly in field trip using the low-level function that's called FT inverse Iloretto. I think that now Nicolas can show you the steps of the code.
Okay, so back to the code. So yes, so using FT prepare lead field, you can compute the lead field. Actually, that was a bad idea. It takes some time, but whatever. Yeah. So what it does, what was done? Yeah, computing it. So it takes. So
okay.
as you see, we have like a bit more than 5,000 sources. So the more sources you have, the more, let's say, the higher the resolution of the reconstruction, but also more computationally heavy it is. And it's also important, though, for the final step to have enough sources, because at the final step, we will assign each sources to a region of interest. So if you have a very sparse representation of your source, some array won't have sources, and therefore then you can't really do any correspondence between patients, because you may miss some array. So it's important to have enough source points to have to be able to reconstruct all the arrays you need. Okay, I was hoping that it was finished while saying that, but it's not. Yeah, so the thing is that the lead field can be wrong because it's a model. So it's always good to verify that the lead field is correct. And to do so, you just represent how one single source can be or projects to the Scalp level. And again, here we have, for each sources, we have 3 orientation of a small dipole. So here it's what I will show you right now. And using this representation, you see that you have the maps. So I hope you see, like, oh, down there, there is the source. This source is pointing towards the right. And you see that you have a nice, smooth electric field. So very centered here and a bit more, yeah, wide on the other side, because it's farther away. And if you change the orientation, also you see it's very smooth, very nice. You have the 2 poles of the dipole. And again, here, if it's oriented in the z-axis, you have 2 nice dipoles. Again, if you show that with sources which is a bit deeper, or a bit more central, you see that you have the same type of topography, but obviously the amplitude level is lower because it's deeper, and we have the same, the source has the same amplitude, but obviously because it's deeper, you have less amplitude at the Scalp. And yeah, the same for all the orientation. Okay, so again, before I mentioned that we had an issue with the BEM model, and you can actually verify that very easily, that it didn't manage to compute a proper lead field, because when you do the same analysis but with this model, you directly see that those topography don't make any sense. And this is very important to do before doing any further analysis, to verify that you have a smooth map. And while here, well, it's completely crap, let's say, it didn't manage to compute a proper lead field. And if you have something like that, it could happen on normal MRI, because again, as I said, you have holes in the mesh, you have something is cut, or you don't have enough space between the layers, and it's a bit difficult to solve. So here it's the BEMCP method implemented inside field trip, and it is known to be a bit, to fail sometime, and it's difficult to debug it. That's why I would highly recommend to use the OpenMeg lead field in head model. It's better because this is not a concentric spherical model, but it's actually doing a so a boundary element model. And you see that this one managed to have a good, nice head model with a realistic head model, and the lead field is correct. And we see those two dipole, and the same here. All right, and now I can show you some results with the Eeg Loretta. So basically, yeah, you use, as Isotta said, the FT inverse Eeg Loretta, and you must also include the lambda for the regularization. And if you do that and you plot this on top of the anatomical MRI, so again, this is also implemented in field trip, you see this, and remember we are doing some visual evoked potential, and you see that it's very so to the posterior part, and especially a fusiform gyrus, and so that's what is expected. Also, I would recommend you to just play around with the lambda and so on, because the lambda is the amount of regularization. So the higher it is, the smoother the results, but also the more leakage you have, which is a problem when you do connectivity. And the lower, less regularization. But then if you have a too small lambda, you do not manage to solve the inverse problem correctly, and then the result is crap, and you see it directly when plotting the traces on the power on the MRI. So final step, Isotta will talk about how we can get the ROI traces.
Okay, so this is the last step. Now we have a signal for each of the sources that we imposed inside the gray matter. We need to go from, actually we have 3 signals for each of these sources within the gray matter. We need to go from this 3D time series over 5,000 sources to 1 time series for each region of interest that can range from 72 to, I don't know, 500, I don't know, ROIs. Of course, in order to do this, we need the parcellation of the gray matter, which is given by the atlas outputted from the connector mapper. We need the spatial filters, so the inverse operator we just calculated in the step before, and then of course we need our clean functional data. So in order to summarize this 3-dimensional information, first of all we import the atlas, and again we set the basal ganglia ROI to 0. Then we need to basically label each of the sources with the corresponding ROI label. And in order to do this, basically for each source point, we find 26 closest voxels around it, and we assign the label of the most present tissue around the source point to the source point. This is done with this function that we wrote, and you can find in the script. And then we concatenate the time courses, because of course at the beginning we epoched it, and here we reconcatenate all the time courses. And for each ROI, we use the singular value decomposition to extract the time series that describes most of the variability of the data we are looking at. So the equation for the singular value decomposition is this, where X are our data, U are the left singular vectors, S are the singular values, and V are the right singular vector. So after we concatenate the spatial filters of the given ROI, and this is done here, we can apply the inverse transformation. So we basically multiply our time courses concatenated by the current filters and by the inverse filters, and then we simply we apply the singular value decomposition, and we store the first left singular value. So sorry, this is the first left singular vector. So the time series that represent most of the variability of the data within that ROIs. And then, of course, once we have done this for each of our ROI, we resegment the ROI time courses in the original epochs, and we're done. And Nicolas will show you.
Okay. And so the final step, again, as she said, so we have to assign a ROI to each sources, then extract them. Here you see we use a path for, so it's a parallel for loop, just to, yeah, it's a bit nice to have this because it shows you how to parallelize this computation, because it's each of the steps are independent. So that's more for common knowledge. It's not, you could use that with a simple for loop. Okay, then once we have them, we basically average each of them according to each trial. Yeah, maybe one thing that is quite important is why do we concatenate all the traces? Because actually what we want is one single decomposition for the DRI, because imagine if you do it for each trial, you have a decomposition, so a space which is different for each trial. And then you don't know really if anything makes sense, because the trials are noisy, and if you have a different orientation and then project that to a given orientation, it may not make sense. Second reason is that even if you find the same subspace, the SVD has a sign ambiguity. So basically you can have one thing, so the X, so let's say here, could become this. So it could change its sign and become something like this. And then it means that between the trials, it will swap the sign, and then it doesn't make, again, any sense to do it like that. So really, you want to have one subspace for the whole recording. Okay, so let's plot that in 3D. So what I do here is I create a mesh for each ROI. Okay, let's do that. Yep, here you have it. So right hemisphere is on the right, left is on the left, and you have the traces of each ROIs. Now we can compute it, and basically what I will do is I will show some specific time point and highlight the region which are more activated at that time point. And should arrive here. So yes, you see it. You have basically the posterior part, then fusiform gyrus going increasing. Here you have the fusiform gyrus with max activity, and then posterior, and then so on. All right. And finally, what you can do if you want is to also plot the ERPs at the ROI level and compare the two tasks, so the face and the scramble task. And you see that you have a big response, for instance, here in the right fusiform gyrus, and this is expected. Yeah, so the blue is the face and the red the scramble. And that's it. And basically, we store, yes, we store the all the traces from the ROIs and all the trials. So that's afterwards. Maria tomorrow will show you how, using that, you can apply, you can use the time variant connectivity to estimate the connectivity across time for this task. And I think
yes. Shall we just say that we've shown a live script loading the results that we obtained separately with 4 separate scripts. So if you want to run it yourself, you should have the scripts in the Git, right?
No,
you can pull it from the Git.
It's already in the virtual machine.
Okay, yeah. So Here you have the EEG preprocessing script, which will give you the finally cleaned EEG data, the MRI preprocessing script, with which you can get the head model and source model estimation. Then the source modeling script, where you get the lead field matrix and inverse operator, and then the ROI time courses, where we summarize the information from sources to ROI. So we recommend you to run those separately and yeah, maybe change some parameters to see how that affect the results. And we did it in time. Incredible. If there is any questions.
Yeah,